Hi everyone, my name is Matthew Nirnett, and just a special thank you for those taking the time to listen to my presentation today. Just to show you who I am, I've got my camera on, but I am going to take it off just so that my slides can display better. The study I'm presenting today is the temporal and spatial ecology of a labiobarba species, particularly the scaly or KZN Yellowfish, in a socioeconomically important river, the Mgeni River. And just a special thank you to the many sponsors that were able to make this study possible. So just an overview of the study. We'll go through an introduction, some of the methods we used, like the fish track program, the results, and then just some concluding remarks. So what's been in the news a lot lately is how poorly managed our river systems are, and primarily because there's been a lot of poor water quality issues and resulting in a lot of fish kills. This was a couple of news headlines taken during the study, and what we're trying to do with the study is to try to prevent these mass kill-offs from happening by understanding the biology and ecology of the species in more detail. This was just a more recent study that highlighted the importance of doing this type of work. So fish are often used as ecological indicators and on different levels of biological organization. And that stems from organism to individual to population. And we've got various tools in which we can assess this across the range of how these tools show their response and sensitivity, as well as the ecological relevance within the organism level, with behavior often being a good medium between being responsive and sensitive towards an issue, as well as ecologically relevant. So we use this within our program, and particularly looking at the behavior of yellowfish, and we wanted to unpack some of their home range, spatial movements, their cues, as well as understand their deal behavior patterns and use of different habitats in light of anthropogenic stresses that are occurring in the system. We chose the fish track program, which is a recently developed fish telemetry technique, and this makes use of both manual monitoring fish telemetry techniques as well as remote monitoring techniques. It also includes a element of monitoring the river condition in real time using probes, and then also telemetry tags attached to the fish within the environment. We're then able to take this data and push it through some data analysis, where we can then determine fish response, some biology and ecology components. But then there's also an element that we can measure this in real time and push this through to how managers manage the environment. So part of the study was to deploy water quality probes within the river, and these probes give us temperature readings, conductivity, as well as the discharge, which is actually recorded as pressure and converted to cubic meters per second. And this is done by use of a hydraulic cross-section with the probe benchmarked onto that cross-section. In addition, we tag fish with telemeter tags, and these tags give us temperature and activity readings. Important component to the study is the fish tag, and has two sensors attached to it. First is the omnidirectional tilt vibration sensor, which records the fish behavior as activity in movement counts per specific time interval, and we chose 1 hour for this study. It also has a temperature sensor which records the water temperature associated with the fish. Data from this tag is stored and then transmitted to the nearest station every 15 minutes. So another important element is to set up a detection network, which consists of a station that has a solar panel on it, as well as an antenna. And these are scattered throughout the the study area and are able to pick up the tags or fish, as well as the water quality probes, and it relays information between the tag and the station, and then stores the data coming off these sensors on a data management system. Our study site was chosen on the Ngeni River in KwaZulu-Natal, South Africa, and primarily between Midmar Dam and Albert Falls Dam. We set up a telemetry network, which are the green triangles shown in the diagram, as well as 2 water quality probes, one on the Carcliff and one on the Ngeni River system. Our upper limit was Howick Falls, near the town of Howick, and fish were able to freely move in the system, with the exception of a couple of in-stream barriers such as Morton's Drift, Carcliff Weir, and Fish Jump Falls. We tagged fish around the Carcliff confluence, as well as the inlet to Albert Falls Dam. These are an example of the scaly or KZN Yellowfish and some of the fish that we had tagged in the river. We tagged up to about 44 fish in total. Here's some of the results. We tagged around 44 yellowfish, and we monitored them from the 9th of August 2018 to the 28th of August 2019. 8 of these were taken out by predators such as otters and fish eagles. And what we found with the home ranges is that the tagged individuals showed high site fidelity, and in at least 33 of these individuals, seldom moving out of their tagging site. As such, there was a high individual plasticity between tagged individuals. Some of the deal behavior of yellowfish showed that they were very active fish, particularly during the daylight periods. What was also interesting is that we found distinct deal patterns between dam and river habitat types, and this was consistent with some other findings where yellowfish actually are serial spawners and will spawn both during the day and night, particularly during summer when they are in the river. So with that, some of the spatial movements that we found was that yellowfish moved out of the dam environment into the river environment during their spawning period, which was predominantly from anywhere from August, September through to April, May of the year. What we found is that they tend to seek out deeper pools for refugia habitat during the cooler, colder months, and then move into the streams during the warmer months. To look at what was cueing this movement up and downstream of the river, we looked at the 11 fish that did move within the study area. And upstream was primarily cued by temperature. During the spring and September, as water temperatures increased, and then downstream movement was primarily cued by the decrease in temperatures in autumn. What we did find is that base flows were relatively kept around 5 cubic meters per second, where fish didn't move as a result of any changes or increases in flow during that period, but did move because there was a change in temperature. When flows had increased late summer, the fish had then moved with the flows and the decreasing in temperatures back into their refugia habitat. And what we determined through this was that access to habitat for spawning is largely provided by the flows, where temperature cues these fish to move. What we also unpacked a little bit, and it's not in the paper that I'm presenting today, but it's interesting results, is we're looking at how these fish respond to the different environmental variables such as temperature, where there's a distinct increase in activity as temperature increases, and interestingly enough, there's a decrease in activity as discharge increases. Electrical conductivity at this stage shows no distinct results. So with this, we found that environmental flows are important to maintain the biological processes for yellowfish, in that they provide access to habitat and provide habitat for yellowfish to spawn, particularly during the high flow season, which occurs during the summer period. And then also we need to improve the way we manage our base flows and our flood releases within this highly regulated river. And there's possible suggestions around that. Just an example is we did do an environmental flow release during the study period, and to see how the fish responded. And this we could monitor in real time, and we could actually see that there was a distinct behavioral change when the flows in the environmental flow release happened. But what we do realize too is that this environmental flow acts as a flush, but it doesn't alter the base flows. And what we need to start looking at is how we manage these and release these environmental flows to maintain biological processes of yellowfish. In-stream barriers did limit the movement of our fish, and so one of the proposals is that any removal of redundant weirs or dams that are no longer used for the purposes that they were initially built for, or not being used at all anymore. And then also, if they are still being used, to retrofit this with in-stream barrier, with fishways, so that we can improve the connectivity and allow access for yellowfish to habitats. The fish track program was a success in the study, and there's a lot more to it that we can, and a lot more that we can use out of it. And we can use the biological response to anthropogenic stresses and use that in real time. And this is just an example of the data that we have, and you can actually see when the fish started to move, and also when their activity increased or decreased during the study. So just in conclusion, every few hours something occasionally happens, but mostly it's just this. And so this is the general perception, but we need to move towards a perception like this, where we understand the behavioral changes and what this means towards management. I'd just like to thank you all for listening to the talk, and again, just acknowledge my sponsors. Thank you.